Hey, welcome back to another Bio 100. Today we're going to start off with just asking this question. If you could swap genes between any organism that you wanted, what would you make? I want you to just hit the pause button and think super broadly. What would you make if you could swap genes between organisms? Okay, hopefully you're back. Hopefully you took some time to think of it. If you didn't, here's some clues. Is this helpful? Hit the pause button again. What kind of organism would you make if you could swap genes between any organisms you wanted? Okay, hopefully you've thought about that a little bit, and that kind of is going to form the focus of our next two lectures. We're going to talk about transcription, translation. We're also going to talk about bioengineering, and hopefully you'll enjoy some of the things that we talk about. Okay, last question. If you wanted to have superpowers, what genes would you need to achieve your chosen superpower? Let's keep this one just a little bit real, but think about it. Okay, hopefully you've thought about it quite a bit, and let's keep rolling. So genetic engineering, right? So there's I'll give you some common examples and then maybe some crazy examples. So one is vaccines. So vaccines are literally just engineered, weakened generally viruses that are injected that prop up your immune system, that cause an immune response without making you sick. Here's another one, crops. So these are genetically engineered for pest protection. So there's all sorts of things that can be done with crops to make them less susceptible to different funguses, even insect pests, etc. And there's a lot of debate around whether this is a good idea or a bad idea. Glowfish. These are literally fish that glow. They can be purchased in 3 stunning colors, as this advertisement goes. But essentially what they've done here is they've put genes into these fish that make them glow in different colors. Now, when I was a kid and I would go to the pet store, they would sometimes paint the fish these glowing colors with nail polish. But these days, they go much beyond that, and they actually just make the fish glow genetically. And then here's one here. They used the genetic sequence of a rat, and they manipulated it to grow a human ear on its back. And these ears the idea is that these ears could be removed and grafted onto a human as well. So that's pretty funky. And there's a lot of engineering going on here in this rat when you look at it. It's also hairless, right? There's a lot happening here. Just don't get sucked in too much. I mean, I know it's hard to stop staring at the human ear on the back of this thing, but it's a lot to think of. And for those of you who might have questions, that is just the cartilage portion of the ear with skin over the top. There's not like an inner hearing portion of that ear. So kind of what we're going to talk about today is how genes and proteins are made and expressed. So protein synthesis and gene expression is what we call this. So from the DNA, what happens is you start with DNA and you go to a gene and then you go to a protein. So protein synthesis is really just the process of using instructions that are carried on a gene to create proteins. And there are several steps involved, and they require both DNA and RNA. So you have the gene, which is the sequence of DNA that encodes for a protein. We've talked about this, right? Genes and alleles and all these different things. And then the protein is just a large molecule that's composed of amino acids that results from the gene sequence. So we've talked about DNA and RNA already, but just a quick refresher, RNA is the ribonucleic acid. It's single-stranded. DNA is double-stranded, remember. RNA, the nucleotides are made of ribose, phosphate, and nitrogenous bases. So DNA is deoxyribose, right? RNA is just ribose. And there are 4 bases. There's an A, C, G, which are all very similar with DNA. But for RNA, there's a U, which is for uracil. And do you know which of the base pairs it replaces? Think about it. It replaces T. So here's what you have. Double-stranded DNA, single-stranded RNA. You'll notice that there's no T's in this string. Those T's have been replaced by U's. And if you're looking across, right, C and C, U, C, you can see how they've been swapped out. Okay. Yeah, and I've kind of been over all this. You can just hit the pause button if you want to read through this to make sure that it's very clear. Okay, so I want you to take a second in your mind. Hopefully you've done your reading. Take a second and define transcription. Okay, take another second and define translation in a biological sense. And if you didn't get those, because maybe you didn't get a chance to do your reading yet or whatever it might be, I'm going to show you this as a hint. And for those of you who are LDS, you should get this right away. And I realize this is kind of a strange hint, but if you think about it, it actually makes a lot of sense, right? So you've got the transcription of the Book of Mormon that happened first, right? And then the translation of the Book of Mormon that came second. And biology is actually the same. Things are transcribed first and then translated later. And here's kind of how it works. Okay, so the flow of the genetic information goes from DNA to RNA to protein. And you can see this down below, starting here with the DNA, right? And then you get a RNA single-stranded transcription of the DNA. And then as you come down here, you're going to get a protein that is translated from the transcribed RNA. So transcription, making RNA, this is going to happen inside of the nucleus. And then the mRNA, once transcribed, is going to pass out into the cytoplasm of the cell. And that's where translation will happen, making proteins. Transcription in the nucleus, as we just talked about, so RNA polymerase is going to bind to the promoter region, right? So here's your RNA polymerase. It's going to bind to what we call a promoter region of the gene. So the promoter region is just a region up of the actual translated portion that tells the RNA polymerase where to sit down. So it will sit down, and then the RNA polymerase will literally zip down the length of the gene, and it's going to match RNA nucleotides with complementary DNA nucleotides, substituting U's in the place of T's. What I want you to do is just take a second, hit the pause button, and look at this and make sure you understand the different pieces that are happening here. Okay, if you don't understand anything that's happening here, it's probably a good idea to reach out to your TAs during some office hours and see if you can really wrap your head around it. Okay, but the product of transcription is messenger RNA, or mRNA is what we call it. And you can see it coming out here, right? So this is the mRNA. Okay, so translation, RNA to protein, where does this happen and what does it require? Go ahead and hit the pause button, see if you can figure that out. So it happens in the cytoplasm, outside the nucleus. And what does it require? It requires mRNA. So this is made during transcription, as we just covered. It requires amino acids, energy. You have ATP that needs to put all this together. And then helper molecules. So you have a ribosome and a transfer RNA. So ribosome is composed of two pieces. You have the large subunit and the small subunit. And those work together to essentially straddle. They run the DNA through here, through the central region, and assemble RNAs. So transfer RNAs, these are what carry the amino acids, okay? And they are going to match in anticodons with the codons of the RNA. And this is all found in the cytoplasm. And I apologize, I think I may have mentioned they ran the DNA through here, right? But they're actually going to run the mRNA. And the result is going to be protein. Okay, so here's how it starts. So you have your amino acids and your transfer RNAs that are really just floating freely in the cytoplasm. You have enzymes, remember we covered enzymes before, that facilitate the binding of specific transfer RNAs to its appropriate amino acid. Okay, so these amino acids are going to be bound to these transfer RNAs according to these different sequences are match up with a certain amino acid. Okay, so the transfer RNA is going to dock if the complementary RNA codon is present on the ribosome. Okay, so here you have it coming in. The protein has docked onto this transfer RNA. It's going to come in and it's going to dock in here according to what's happening here, right? So this is an AGG, so it should match up with a UCC, right? That's going to come in, it's going to dock, and then these are going to be, the amino acids are going to be joined together to form a polypeptide. Okay, and as that continues, the ribosome is just going to keep moving on to the next codon. It's going to keep receiving different transfer RNAs. Those transfer RNAs are going to all get combined together into this polypeptide until they reach what's called a stop codon. And a stop codon has no transfer RNA base that can land on there, and so it actually terminates the end of the newly synthesized protein, and it will be released. Okay, and then the amino acid folds, and the protein is ready to perform whatever job it's been asked to do. The subunits of the ribosome are then just going to separate. They can reassemble and begin translation on another mRNA that comes from the nucleus. So I want to just run you through a video of this real quick, and hopefully you enjoy this.
The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific three-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
So hopefully you enjoyed that and that was insightful. But just think about this, right? 100 trillion molecules per second of hemoglobin that are being made in your body, which means that just in the time that you sat there and watched that clip, literally hundreds and hundreds and hundreds of trillions of proteins have been produced in your body through transcription and translation, which is pretty cool to think about. Okay, so the genetic code. We've talked a little bit about this as we've come up against it. So the genetic code really has this specific codon to code for a specific amino acid. So you have a codon is comprised of 3 nucleotides, and there are 64 possible combinations, right? 4 to the third combinations, makes sense? 61 codons code for amino acids, so 20 total amino acids, but 3 others are what we call the stop codons, which we talked about already, which end protein synthesis. So if you're looking at it here, so essentially a UUUU, 3 U's, a UUC, these are going to make phenylalanine. Jump over here, you have these 4 combinations of nucleotides are going to make arginine, okay? And arginine is found in multiple, so there's actually 6 combinations for arginine, right? And you can look across here and see what makes valine, alanine, right, proline. And then you have your stop codons here, and then your start codons. So this is where the protein knows to begin to be made. Okay, and as we mentioned, specific codons are going to code for specific amino acids. You can hit the pause button here and spend a lot of time looking at this if you need to. But here's what I want to do. Given the DNA sequence, I'm just not going to read it off, but of this DNA sequence right here, what is the amino acid content of this protein? Using this table and this sequence, you should be able to do transcription and translation and get a protein. So hit the pause button, get out a piece of paper or some sort of device that you can write on and work this out. I'm just going to hang back for a minute. Okay, hopefully you actually took the time to do that. If you didn't, I highly recommend that you hit the pause button and do it. But this is what you should have gotten. Okay, so if your DNA is going to be AAT, TT, CCT, GAT, ATT, it should result in an RNA molecule that is complementary to that, but with T's being substituted, with U's being substituted for the T's. So essentially you should have UUA, AAA, GGA, CUA, UAA. Okay, and then the subsequent protein should be leucine, lysine, glycine, leucine, and then it should end in a stop codon there. All right, mutations. So where are changes in the genetic sequence? Changes in the genetic sequence are called mutations. We know this, right? But do all DNA changes affect the order of amino acids? Meaning, could you have a mutation from what you've seen in that previous table? Can you have a mutation and change amino acids? So changes in the genetic sequence might, affect the order of amino acids in a protein. And why is it just a might? When would it and when would it not? And hint, right, protein function is really dependent on the precise order of amino acids. Okay, and before we answer that question, let's come back and do this and look at this. So what are the possible outcomes of mutation to a protein? Okay, so one, there can be no change in a protein, meaning that there's a neutral mutation. And you can look down here, right? So if the sequence is a UCU and there's a mutation in the sequence where you get UCC, it doesn't change the protein at all. So the protein, we call these neutral mutations. They don't change anything. They can also result in a non-functional protein where, let's say that this UCCU is actually mutated to a UUU. That would be phenylalanine instead of serine, and that could cause big trouble. It could also result in a different protein. If you, let's say that the phenylalanine substitution for serine is actually still something functional and works, it just means you might have a different shaped protein. So here are some of the mutations that we can look at. Okay, and I want you to just keep in order here. These are all organized in threes here. So this is your normal gene. Okay. So if we get a substitution change here, if we get a C in here instead of a T, it can actually change the protein to a valine. If you get what we call an insertion or a deletion, in this case they're showing an insertion where a base pair is inserted, it can actually shift all the proteins downstream. And the same thing can happen if you erased essentially or lost an amino acid, a base pair in here. It can change your protein downstream, and this is called a frame shift. Here are some others here, right? So this is your nice, beautiful looking protein. If there's no substitution, but if you get a mutation in here that changes that to a glycine, you can get a completely different protein. And this protein is actually non-functional. You can see there's a receptor site in here. This can latch on to something to make it functional. That site is not present in this one. Okay, and then they're what we call neutral frame shifts, right? So you can actually have something where you get a mutation and the amino acid is going to be the same as the original, as we talked about a little bit ago. And then you can have another one here where you get this insertion, and that's going to lead to a stop codon. It's actually going to cut the protein off, so the protein doesn't even have all of the pieces that it needs to be functional. All right, and we actually see this in humans even. Okay, so sickle cell anemia. I'm not sure how many of you know much about sickle cell anemia, but you should definitely take a few seconds and search it out and understand it. It affects human populations, specifically many human populations with African origins. And what happens is you have this single change here where instead of getting this healthy looking quote-unquote normal hemoglobin here with the protein glutamate, you actually get what we call sickle cell shaped hemoglobin because you have a valine. Okay, so we're not just like making up stories and drawing stuff up, like this really happens. So it's in the hemoglobin gene, it's really just a one base pair change, and it equals an amino acid change, which brings in the sickle cell phenotype. For those of you who took some time to look up what sickle cell anemia is, you probably figured out that it is the hypothesis is that it is in relation to malaria, and individuals that have sickle cell anemia are immune to malaria, but it comes with many other downsides, meaning that generally individuals with sickle cell anemia don't live as long, although they live long enough to pass on their genes. Okay, and then let's kind of go into an overview of gene expression. So each cell in your body, except sperm and egg cells, have the same DNA. And you should understand why sperm and egg cells do not have the exact same DNA, right? It's because they have half of the DNA. than the rest of the cells in your body. But each cell is only going to express a small percentage of genes. So for example, nerves and muscle cells are going to perform very different functions, so they're going to have very different genes. How does this all happen? Well, turning a gene or a set of genes on and off is how this happens, and it's called regulating gene expression. Okay, and so if you look at this, they have very different even structures. The cells themselves are structured very differently, and this is just from expressing different gene contents. All right, so let's jump in here real quick. I want you to hit the pause button and look at this and see if you can make sense of anything that's going on in this slide. All right, if you were able to make sense of that, great. If you weren't, don't worry, we'll talk about it next time. And that's all I have for you today, and I'm looking forward to our next lecture.